This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome. This narration explores one of paleontology's most extraordinary windows into deep time, the Dengying Formation of South China, a sequence of ancient rocks preserving life on Earth during its final Ediacaran chapter. Across 5 chapters, we'll journey through remarkable fossil discoveries, examine the chemical secrets locked within half-billion-year-old limestone, and confront a fundamental question. When did the animal world we recognize truly begin? From iconic Ediacaran creatures to the earliest known relatives of our own evolutionary lineage, the story the Dengying Formation tells is stranger, richer, and more consequential than anyone anticipated. Chapter 1. At the Threshold of the Cambrian. Deep in the sedimentary archives of South China, roughly 500 million years of geological time are locked within a series of rock layers that have come to represent one of paleontology's most important and productive hunting grounds. This is the Dengying Formation, an upper Ediacaran sequence stretching across several provinces of South China, and it sits at a position in time that scientists have increasingly recognized as truly pivotal. The formation spans from approximately 551 million years ago to 538.8 million years ago, a window of just over 12 million years pressed into a thickness of carbonate-rich rock that sometimes reaches hundreds of meters. But what makes those 12 million years so extraordinary is not their duration so much as their timing. The Dengying Formation captures life on Earth during its final Ediacaran chapter, a period when the biological world was about to undergo one of its most dramatic transformations. The Ediacaran period itself ran from about 635 million years ago until the dawn of the Cambrian. During most of that interval, Earth's oceans were populated by soft-bodied organisms unlike almost anything alive today. Frond-like creatures anchored themselves to the seafloor and filtered nutrients from the water. Disc-shaped organisms lay against microbial mats. Tubular forms rose and sprawled across the ancient seabed. Then, at the close of the Ediacaran, something shifted. The biological recipe changed, and the ocean floor began filling with the ancestors of animals we recognize. Creatures with heads and tails, bilateral symmetry, feeding structures, and a capacity to move with purpose. The Dengying Formation documents this shift from the inside, preserving fossils from across that critical transition in a variety of rock types and environments spread across a vast geographic area. The formation's distribution across South China speaks to the scale of the ancient shallow marine platform that once dominated this region. Different provinces preserve different rock sequences and different members within the formation, each with its own character and fossil content. In Hubei province, the formation overlies the earlier Dushantua Formation, and its members, particularly one called the Shibantan member, consists of dark, thin-bedded bituminous limestone deposited in a subtidal environment. The Shibantan member is among the most fossiliferous units in the entire formation, holding a remarkable assemblage of organisms that straddles what paleontologists call the White Sea and Nama assemblages of Ediacaran life. In Shanxi province, the Gaojiashan member contains its own distinctive fauna within a sequence that records substantial environmental fluctuations, including episodes of marine anoxia. In Yunnan province, the Jucheng member sits within a mostly siltstony sequence and has become the source of some of the most startling recent finds. And in Sichuan, the Dengying Formation is best known to petroleum geologists rather than paleontologists, since its ancient carbonate platform facies, microbial mounds, and grain shoals built up over millions of years now contain enormous reserves of natural gas deep beneath the basin. That duality of scientific interest reflects the unusual completeness with which ancient, shallow carbonate platforms can preserve records of both biology and environment simultaneously.
The carbon, sulfur, and strontium isotopes locked within these limestones and dollostones carry a detailed record of the terminal Ediacaran ocean's chemistry, one examined at length later in this book. But the fossil contents of the formation demand attention in their own right. The animals and algae preserved within the Dengying Formation reflect the ecological complexity of a world in flux. The Shibatond amber alone preserves frondose organisms that belong to groups familiar from much older Ediacaran sites elsewhere in the world. Charnia, that iconic frond-shaped organism whose discovery in Charnwood Forest in England in 1956 and 1957 first convinced the scientific community that complex life existed before the Cambrian, appears here. So does Charnia discus, Pteridinium, and Rangia, a roster of Ediacaran organisms that researchers have debated for decades in terms of their relationships to modern animal groups. The fact that these organisms persist into the terminal Ediacaran, preserved now in carbonate rather than sandstone, has important implications for understanding how widely distributed and environmentally tolerant they were. For a long time, most Ediacaran fossils were known only from sandstone and siltstone environments. So finding classic Ediacaran forms preserved in marine limestone significantly expanded scientific understanding of where these creatures actually lived.
Perhaps one of the most unexpected finds from the Shibantan member has been the presence of Dickinsonia, one of the most recognizable and studied of all Ediacaran organisms. Oval to elongated, covered in a quilted pattern of segments, and widely interpreted as a mobile organism capable of moving across the seafloor, Dickinsonia had previously been known almost exclusively from siliciclastic deposits in Australia, the Baltic region, and possibly India. Its discovery in the limestone facies of the Shibantan member extended its known geographic and environmental range considerably, hinting that Dickinsonia was more cosmopolitan and ecologically flexible than previous evidence had suggested. Only a single incomplete specimen has been described from the Dengying Formation, recovered from approximately 1 meter above the base of the Shibantan member at the Wuhei Quarry, but its presence alone raises intriguing questions about the extent to which Ediacaran organisms had spread across the ancient world. Beyond the fronds and discs, the Dengying Formation also preserves a variety of tubular organisms that represent some of the earliest experiments in constructing mineralized or semi-mineralized body coverings. Claudina, a small tube-dwelling animal whose nested funnel-like construction is among the oldest evidence of animal biomineralization anywhere on Earth, appears in the Gaugiashan member as well as in the Shibantan member. Alongside it, organisms like Conotubus, Gaugiashania, and Cinotubulites built their own tubular homes in the seafloor sediments. These fossils have fascinated researchers precisely because they document the very beginnings of hard part construction in the animal kingdom, a biological capability that would eventually transform the planet's fossil record and is closely linked to the spectacular diversity of the Cambrian that followed. The Gaugiashan Lagerstätte, situated within the Gaugiashan member of Shanxi Province, has been the focus of intensive geological and geochemical investigation. This site is notable for pyrite-dominated preservation of its tubular fauna, particularly detailed studies of how organisms like Gaugiashania cyclus constructed their annulated tubes and interacted with their environment. Overlying the Gaugiashan member in the Shanxi sequence, the Beiwan member adds its own contribution to the formation's tubular fauna, with organisms including Paleoropilon spiniferum and Schufongtubulus inornatus, extending the diversity of tubular constructors into the formation's later stages. The stratigraphic position of the Beiwan member places it at or possibly just across the Ediacaran-Cambrian boundary, an ambiguity that points to a broader uncertainty about where the Dengying Formation actually ends. Traditionally, the uppermost unit in many South China sections was taken to be the Tianzhushan member, but this member shares sedimentological and geochemical characteristics with the overlying Cambrian Yanjiehe Formation, blurring the line between the two systems. The final pages of the Dengying Formation's record may, in places, already belong to the Cambrian world that is supposed to succeed it, a complication that researchers working on this boundary continue to navigate. Then there is the Tongshan Lagerstätte, located in Hubei province and formally described in late 2025. This site preserves its organisms through Burgess shale-type carbonaceous compression, a style far more commonly associated with the Cambrian, making it a genuine outlier among Ediacaran localities. Its assemblage includes the 8-armed Eoandromeda octobrachiata alongside the first known rangeomorph fronds preserved with this quality of taphonomic fidelity. What that exceptional preservation mode reveals about Ediacaran organisms and the communities they inhabited is explored in full in a later chapter. The Yunnan province section of the Dengying Formation introduces an entirely different flavor of discovery. The Zhucheng member has yielded a collection of organisms that blur the boundary between Ediacaran and Cambrian life in ways that have left researchers re-examining long-held assumptions. Among the known forms from this member is Lobodiscus tribrachialis, a triradial disc-shaped organism belonging to the Trilobozoans, alongside others that resist easy classification as either Ediacaran or Cambrian in character. Within this same member, the Zhangchuan biota, reported in a 2026 study, has yielded an assemblage whose full significance is the subject of the chapter that follows.
Across its several members and localities, the Dengying Formation accumulates into something greater than a catalog of late Ediacaran life. Helicolicellus cantori, a crown group sponge from the Shibantan member, the bilaterian Yelingia spiciformis, whose distinctive movement traces cross the ancient seafloor, looping burrows attributed to Lamonte travalis, documenting behavioral complexity, and possible lobopodian trackways in the Shibantan member, suggesting organized locomotion by articulated animals. These details, drawn from different rock types and different localities within the formation, collectively sketch an ecosystem of remarkable sophistication. Whether the Dengying Formation's organisms were Ediacaran holdovers, Cambrian pioneers, or creatures that defy both categories is a question the fossil record keeps forcing researchers to reconsider, and one that the Jiangchuan biota, in particular, has made considerably harder to answer with confidence. Chapter 2. When Old and New Shared the Sea. The team from Yunnan University and Oxford University had not come to the hills above Fuxian Lake in Yunnan province looking for what they found. The locality was already known to researchers, but for its algae, Ediacaran plant-leg organisms whose carbonaceous remains had made the site a productive destination for those interested in pre-Cambrian primary producers. It was in pursuit of those organisms that survey of the Zhucheng member of the Dengying Formation began. What emerged from the rock instead was an animal assemblage unlike anything previously recovered from rocks of this age. It was unlike anything I've ever seen, said co-author Francis Dunn. Fan Wei, who led the fieldwork, described the find in terms of years of searching. After years of fieldwork, we finally found several sites with the right conditions where animal fossils are preserved together with abundant algae. The study, led by Gaorong Li and involving co-authors including Dunn, Luke Perry, Ross Anderson, and Pei-Yun Kong, was published in the journal Science in April 2026 and has since forced a reconsideration of how far back the animal world we recognize as Cambrian actually extends. The Zhangchuan biota, as this assemblage is now formally designated, comes from rock layers positioned approximately 7 million years below the first clearly Cambrian deposits. The fossils are preserved as thin carbonaceous films, sheets of organic carbon pressed flat against fine-grained sediment, a style of preservation far more typical of Cambrian deposits, like the Burgess Shale, than of anything previously known from the Ediacaran period. The rarity of such conditions in Ediacaran rocks makes the Zhangchuan site an exceptional rather than representative window. Fine-grained quiet water settings capable of producing carbonaceous compressions of this quality simply did not leave a consistent record in Ediacaran strata the way they did in the Cambrian.
Over 700 specimens have been recovered, most of them small and rendered as stark black silhouettes against gray rock, yet preserving an unusual degree of anatomical detail, internal organs, feeding structures, and what has been interpreted as muscular tissue.
The most scientifically consequential organisms among them are fossils interpreted as stem group ambulacrarians, the earliest confirmed members of the deuterostome lineage, the great branch of bilaterian animals that would eventually give rise to echinoderms, hemichordates, and through a long chain of evolutionary succession to vertebrates, including our own species. For decades, molecular clock analyses, studies that estimate divergence times by tracking mutation rates across living genomes, had consistently placed the split between deuterostomes and their protostome relatives deep in the Precambrian, with many estimates suggesting origins anywhere from 600 to over 700 million years ago. The Zhengchuan specimens are the first unambiguous fossil confirmation of those predictions, closing a gap between the molecular and paleontological records that had been one of the most conspicuous anomalies in early animal evolution. These organisms resemble animals known from the Cambrian, particularly forms in the Cambro-Urnid and Herpagaster lineages, with bodies set atop stems and tentacle-bearing head regions used for suspension feeding. The Zhengchuan examples are simpler in construction than their Cambrian counterparts, as one would expect of early members of a lineage still at an initial stage of diversification. Several individuals have been identified, and their structural resemblance to later Cambrian forms is close enough to make the lineage assignment persuasive, while their overall simplicity marks them as something genuinely ancestral rather than fully derived.
The significance of finding these organisms in the Ediacaran is sharpened by the baseline against which they stand. Before the Zhengchuan study, as many as 4 individual bilaterian species had been proposed from Ediacaran rocks worldwide, and each one was contested, carrying enough anatomical ambiguity that its bilaterian status remained arguable to a skeptical reader. Trace fossils hinted at the presence of animals capable of directed movement. Certain body fossils had been interpreted as bilateral on structural grounds only to attract counter-interpretations. Against that thin and disputed record, the Zhengchuan assemblage arrives with the force of numbers. The animals that dominate the Zhengchuan community in sheer abundance are a group of vermiform creatures documented from 185 individual specimens in a single sedimentary horizon. Each individual consists of a large circular holdfast disc anchored to the substrate, connected by a flexible stalk to an elongated body with a terminal mouth at the free end. That mouth was apparently capable of everting outward, turning itself inside out to extend feeding structures beyond the body wall, a mechanism familiar from some living annelid and platyhelminth worms. The largest complete examples reach approximately 61 millimeters in length, with a body width of around 2.5 millimeters, while their holdfast discs can reach 15 millimeters in diameter. 185 specimens of a single species crowding an ancient seafloor is not a marginal result. It is evidence of a successful, ecologically dominant population, the kind of argument that no contested individual specimen can make on its own. A related finding concerns another fossil type in the same stratigraphic unit. The disc-shaped Cycleomedusa zhengchuanensis, previously described as a standalone discoid organism, has been identified as possibly representing the detached holdfasts of these worm-like animals, preserved in a different sedimentary layer from the bodies themselves. If correct, this relationship illustrates how easily the parts of a single organism can be misread as separate taxa when preservation separates them across different rock horizons. 6 other specimens in the Zhengchuan biota preserve a cup-like body bearing terminal arm-like extensions, a form immediately recognizable as resembling Haotia quadriformis, a muscular cnidarian known from the Ediacaran of Newfoundland and dated to approximately 560 million years ago. Because the Zhengchuan material is placed at roughly 547 million years ago, these specimens postdate Haotia by approximately 13 million years rather than preceding it. They are a geographically distant and slightly younger relative, smaller than the Newfoundland material, reaching at most 15 millimeters in length and 20 millimeters in width. and sufficiently distinct in detail to potentially warrant description as an entirely new genus alongside Hahootia and its close relative Momcetia. Preserved within their body walls is what appears to be fibrous tissue consistent with musculature, suggesting that active, directed behavior had evolved in Kynetarians well before the Cambrian began, and that muscular Kynetarians had diversified across vast geographic distances by the terminal Ediacaran. A single specimen, roughly 18 millimeters long and 10 millimeters wide, preserves structural rows comparable to the comb rows of cilia-bearing locomotory plates that define the ctenophore body plan today. Ctenophores had been documented in their preserved form only from the Cambrian before this find, with those examples illuminating the early evolution of their characteristic sensory and nervous architecture. This Zhangchuan specimen, compared to the Cambrian form Ctenorhabtatus, would push the confirmed presence of this lineage into the Ediacaran, suggesting that some features previously considered Cambrian innovations were established millions of years earlier. Another single specimen, 18 millimeters long and only 3 millimeters wide, has been compared to the Mackenzides, a poorly understood Cambrian group characterized by rows of internal tube-like structures whose phylogenetic position and lifestyle remain contested. Mackenzides had attracted attention for their seemingly archaic anatomy, which some researchers argued might reflect Ediacaran biological heritage persisting into the Cambrian. The Zhangchuan specimen's modest size, notably smaller than its counterparts from the Chengjiang fauna, may represent an early evolutionary stage of a lineage that would expand and diversify once the Cambrian got underway.
Alongside the deuterostome-like forms, the assemblage contains elongated fossils resembling margarita, a tube-like structure from the Cambrian thought to have served as dwelling tubes for hemichordate worms. These examples feature rows of ovoid holes running along their full length, and the largest complete specimen reaches 30 millimeters in length and 4 millimeters in width. A single sausage-shaped organism in the assemblage preserves a circular terminal opening interpreted as a mouth and a long internal trace interpreted as a digestive gut. A simple but remarkable snapshot of animal anatomy captured across 500 million years. The power of the Zhangchuan biota lies not in any single organism, but in the assemblage as a whole. This is a documented community captured in a single sedimentary layer where organisms from groups traditionally considered Cambrian innovations coexist with recognizably Ediacaran forms, discoid, frondose, and modular organisms whose counterparts populate other Dengying Formation localities. This coexistence directly addresses the long-standing debate about whether Ediacaran and Cambrian faunas were sequential or overlapping in time, and the Zhangchuan evidence strongly favors overlap. Ross Anderson, one of the Oxford co-authors, framed the interpretive stakes directly. Our results indicate that the apparent absence of these complex animal groups from other Ediacaran sites may reflect differences in preservation rather than true biological absence. The broader Jucheng member, deposited in roughly the same time interval, reinforces this picture from a different angle. Its fauna includes multiple forms bearing striking resemblances to Cambrian animals beyond those catalogued from the Zhangchuan assemblage specifically. Organisms that resist easy categorization as either Ediacaran or Cambrian in character. There is the sail-shaped Alienum velaminus, whose body plan echoes that of the Vitilicolians strongly enough to challenge assumptions about when filter-feeding architectures of that kind first appeared. There is a tetraradial organism whose lobes recall both the trilobozoan Tribrachidium and the triradial Lobodiscus, a form whose geometry cuts across what researchers had assumed were discrete taxonomic categories. Together, the Zhangchuan biota and the broader Jucheng fauna constitute a portrait of a biological world already in genuine transition, one where bilateral symmetry, suspension feeding, muscular tissue, and deuterostome body organization were features of a functioning community millions of years before the geological boundary that conventionally marks the Cambrian. The critical question that follows is why assemblages like this are so rarely encountered.
If bilateralians and deuterostomes were present in terminal Ediacaran oceans in populations dense enough to dominate a locality, why does the wider fossil record of the Ediacaran give so little sign of their existence? That question points not toward biology, but toward something far more contingent, and it is what the next chapter examines. Chapter 3. What the Rock Keeps Hidden. The question left standing at the close of the previous chapter was deliberate. If cnidarians, chytnephorores, bilateralians, and the earliest known relatives of deuterostomes were genuinely present in terminal Ediacaran oceans, living in populations dense enough to dominate a community, then why are comparable assemblages not found across the rest of the Ediacaran geological record? The answer begins with a difficult and somewhat humbling realization. A significant portion of what scientists have understood about the timing and pace of early animal evolution is not a straightforward record of biology, but a record shaped, filtered, and in many places silenced altogether by the specific and demanding requirements of fossilization. The Dengying Formation offers a uniquely revealing case study precisely because it contains, within a single formation spread across multiple provinces and rock types, a greater diversity of preservation styles than almost any other Ediacaran unit known. Reading them together reveals both the richness of terminal Ediacaran life and the mechanisms by which so much of it has been systematically hidden from science. The most recently documented of these preservation windows, and in many ways the most conceptually disruptive, is the Tangshan Lagerstätte from Hubei province. What sets Tangshan apart is not merely the organisms it has yielded, but the preservation gradient its assemblage explicitly demonstrates. The researchers working on the site identified 3 distinct levels of fidelity operating simultaneously within the same fossil community. Traditional Ediacara-type preservation that records only surface outlines, a more informative variant that retains organic residues alongside those outlines carrying compositional information invisible in simple impressions, and full Burgess shale-type preservation that captures the structural architecture of soft tissues at a level previously associated almost entirely with Cambrian deposits. The organisms preserved at each level are not distributed randomly across those tiers. They reflect genuine biological differences in tissue composition, structural organization, and the concentration of resistant organic molecules in different body compartments. That correspondence between taphonomic outcome and biological input transforms Tangshan from a curiosity of one unusual locality into a diagnostic framework for interpreting what any Ediacaran preservation mode can and cannot communicate. The metazoan community at Tangshan spans an unexpectedly wide taxonomic range. The sponge Sinospongia chenjunyuani is present alongside Eoandromeda octobrachiata and the rangermore fronds that have drawn the most analytical attention. 2 fan-shaped forms recalling Bradgatia from other global Ediacaran sites are preserved within the assemblage, one of which carries a possible holdfast structure. Organisms with less certain affinities extend the roster further still. Protoconites minor, with a possible cnidarian connection, and Paleopicicnus linearis, with a possible foraminifer affinity, push the community's taxonomic width toward groups that rarely appear with confidence in Ediacaran deposits. Most significantly for what the preservation mode can reveal, the rangermore fronds here achieve Burgess shale-type fidelity for the first time anywhere in the world. One specimen shows 3 distinct longitudinal regions, a central zone flanked by 2 lateral petaloids, with primary branches arranged in parallel and secondary branches oriented nearly perpendicularly to them, all on the same individual, whose body also tapers distally and curves to one side. What had required inference at sites where only impressions survived is here readable directly from carbonaceous residue. Rangermorphs possessed enough organic substance in their bodies to leave chemical traces, with implications for how biochemically substantial these organisms actually were.
Among the tongue-shan organisms, Eoandromeda carries its own preservation lesson. Previously known from deposits dated near 551 million years ago, approximately the boundary between the White Sea and Nama assemblages of Ediacaran life, this 8-armed radially symmetrical organism had appeared to vanish from the record before the Nama assemblage interval began. The Tung-shan locality, bracketed in age between that earlier horizon and approximately 543 million years ago, extends Eoandromeda's confirmed existence into the Nama interval itself. The researchers concluded that this younger occurrence indicates the organism's earlier perceived disappearance was a taphonomic artifact rather than a genuine extinction. It was presumably present throughout the intervening millions of years, but fine-grained organic preserving conditions capable of capturing it simply did not accumulate in the right places during that gap. The evolutionary implication extends beyond this single genus. Apparent extinction events derived from the Ediacaran record must be evaluated with skepticism proportional to the rarity of the preservational conditions that first made a given organism visible at all. The Jiangchuan biota extends this argument in a direction that touches not just the Ediacaran record, but the interpretation of the Cambrian explosion itself. As the preceding chapter details through the researcher's own words, the scarcity of comparable assemblages from other Ediacaran sites most likely reflects differences in preservation rather than true biological absence. The densely populated worm-like fauna documented there represents a community operating at an ecological scale that demands explanation when similar assemblages fail to appear elsewhere in the terminal Ediacaran. If that absence reflects the rarity of carbonaceous compression conditions in Ediacaran strata rather than the rarity of the organisms themselves, then the Cambrian explosion, interpreted for decades as the moment when animal life suddenly and dramatically diversified, may partly be a taphonomic phenomenon. As fine-grained Burgess shale-type deposits became more common in the early Cambrian, they began to document communities that had already been ecologically complex for millions of years. The explosion in the fossil record may partly reflect an explosion in the type of sediment capable of preserving soft-bodied animals, a real signal coinciding with, but not entirely caused by, the biological diversification underway at the same time. That interpretation carries its own ceiling. The environmental and geochemical changes crossing the Precambrian-Cambrian boundary were genuine drivers of biological innovation, not merely noise around a taphonomic signal, and the chapters that follow address what the geochemical record contributes to that remaining gap. Turning from these fine-grained windows to the Shibatan member introduces a different kind of puzzle with its own evolutionary payoff, independent of the preservation chemistry that Chapter 4 examines in detail. The most fundamental contribution of the Shibatan fossils is their setting, frondose Ediacaran organisms, Pyridinium, Rangea, Charniodiscus, preserved within a clearly marine subtidal carbonate. That fact alone dispatches the terrestrial hypothesis for Ediacaran life, associated most prominently with the geologist Greg Retallack, which proposed that classic Ediacaran organisms might represent life on soils or paleosol surfaces rather than the ocean floor. Marine limestone cannot form in terrestrial or freshwater conditions. Whatever these organisms were, at the Shibatan member they were unambiguously marine and unambiguously subtidal, a result that also extends the confirmed habitat range of the Ediacaran assemblage beyond the sandstone and siltstone settings where most of its members were first described. The event beds interspersed through the Shibatan sequence played a dual role in the community they helped preserve. They were burial agents, smothering organisms rapidly enough to initiate the authigenic mineral precipitation that would eventually fix their outlines in rock. But they also imported nutrients, carrying material swept from shallower, more energetically active parts of the surrounding platform into the quieter subtidal basin. The fossil communities associated with these event horizons suggest that organisms were responding to those periodic inputs. What the Shibatan preserves is therefore not a static cross-section of background conditions, but a community partly organized around the episodic supply of resources from a wider platform system.
The joint occurrence of Lamonte travalis and classic Frondos Ediacaran body fossils in the same unit carries evolutionary weight beyond what either record communicates alone. Burrowing bilaterians and Frondos Ediacaran organisms were not sequential here, but simultaneous, sharing the same depositional environment at the same moment in time. This directly challenges the ecological displacement model for Ediacaran extinction, which holds that mobile animals outcompeted or consumed the passive Ediacaran forms that preceded them. At the Shabantan member, displacement is not what the record shows. Whether coexistence was globally representative or captures a local snapshot before displacement began elsewhere is a question this locality alone cannot settle, but it establishes the relationship as a real ecological fact rather than as a consequence of temporal compression in the rock. At Gaozhashan, the pyrite replacement chemistry that Chapter 4 examines in detail yields evolutionary insights that stand independently of the mechanism. What becomes visible through pyrite fidelity is not a single homogeneous biological category, but a spectrum of tube-building strategies, each occupying a different position on the long evolutionary trajectory from purely soft-bodied organisms toward the fully mineralized shells of the Cambrian. Some cynotubulites variants appear to have constructed their walls primarily through incipient biomineralization, secreting mineral matter as a direct biological product, while others relied on organically reinforced sheaths, stiffening their coverings through biochemical organization of existing organic material. Still others appear to have combined both strategies within the same tube wall, producing structures whose material properties resist clean assignment to either category. Conotubus and Gaozhashania cyclus represent distinct solutions to the same underlying challenge of building a persistent external covering. Gaozhashania's annulated construction, with interlocking segments implying growth organized from a defined end, suggests a degree of developmental complexity substantially beyond the simple extrusion of an undifferentiated organic tube. What the Gaozhashania material collectively documents is not a single invention of tube building, but the simultaneous presence of multiple distinct approaches to the problem, tested at a geological moment before any of them had been driven to competitive resolution. The fully mineralized Cambrian shell lay far in the biological future. These tubes represent an intermediate ground, a variety of working experiments rather than a single evolutionary prototype. The vase-shaped microfossils preserved by phosphatization add a further layer to this community picture. Protologena and Cicilogena, tiny flask-shaped organisms among the earliest testate microorganisms in the fossil record, are known from this part of the formation precisely because phosphatization captured their delicate tests before decomposition could erase them. Their presence reveals a protestant community layer beneath the macrofossil record, one that contributed to ecosystem ecology and to the geochemical cycling of nutrients that may have influenced which macroscopic organisms could persist at particular horizons. At the very base of the formation, at the Lianghe section in Zhenba County, phosphatized microfossils interpreted as probable stem group coralline algae make the same preservational principle visible in a botanical direction. The identification rests on morphological specifics. These fossils display columnar thalli and a stromatolithic tissue construction, erect, organized growth in a pattern comparable to modern coralline architecture that provides the anatomical basis for placing them early within that lineage rather than among the broad and poorly differentiated microbial community from which coralline algae ultimately descended. Without phosphate-rich pore waters at burial, these structures would have decayed without trace, and what they document, organized, erect, calcification-capable algal growth tens of millions of years before any previously confirmed coralline occurrence, would remain an inference rather than a fossil fact. Mapping the selectivities of all these preservation modes together reveals not just what has been found, but the specific shape of what the current record cannot access.
The organisms most systematically invisible across all of the Dengying Formation's windows are those with entirely soft, rapidly decaying tissues and no structural organization capable of guiding mineralization. Precisely the organisms that, if present, would most profoundly complicate current models of terminal Ediacaran ecology. Whether such organisms are absent because they had not yet evolved, or absent only from the fossil record because nothing in the formation was built to preserve them, is a question the existing windows cannot answer on their own. Answering it would require a preservation mode not yet documented in the Dengying, one that may already exist somewhere within the hundreds of meters of rock still awaiting the right conditions of exposure and recognition.
Chapter 4, The Chemistry of Memory, Taphonomy, the science of how organisms become fossils, is never a neutral process. Every fossil assemblage is the product of a specific chain of geological and chemical events that began the moment an organism died. And understanding that chain is not merely a technical footnote on how to interpret what is found. It is a direct window into the ancient environments those organisms inhabited, the biological properties that made some of them preservable and rendered others invisible, and the sedimentary conditions that either captured or erased the record of their existence. Within the Dengying Formation, preservation was not uniform but spectacularly varied, and examining how each major Lagerstätte came to exist, the geological settings, the sedimentological dynamics, and the geochemical environments that converged to capture life at each locality, reveals a layer of insight that deepens the biological story the fossils themselves tell. Begin with the geological circumstances that brought the Shubontan member into existence, because those circumstances were causally linked to what the Shubontan preserves. The bituminous limestone of the Shubontan does not represent a continuation of the shallow, energetic carbonate environment that preceded it. Directly below in the Hubei province sequence sits the Hamajing member, light gray, medium to thick-bedded dolomite representing paratidal and sub to supratidal conditions, deposited above the fair weather wave base in a setting periodically exposed to wave energy and occasionally emerging from the water altogether. The transition between these two units is sharp, not gradual, and that sharpness records a geological event rather than a gentle environmental drift. The most likely cause is a tectonically induced increase in subsidence, the seafloor pulling downward faster than carbonate accumulation could keep pace, deepening the depositional environment rapidly enough to shift from the shallow, evaporative dolomite realm to a subtidal basin setting, close to the storm wave base. That abrupt local deepening created the quiet, anoxia-prone, carbonate-accumulating setting in which the Shubontan's remarkable fossil community was able to develop and eventually be preserved. The significance of that depositional position, near but not below the storm wave base, cannot be overstated in taphonomic terms. In quieter waters, microbial mat communities suppressed in the more energetic Hamajing environment could now flourish on the seafloor, creating a biologically active organic layer that both stabilized the substrate and provided a physical surface against which organisms could be pressed and held after death. The same quiet conditions permitted fine-grained carbonate mud to settle slowly and continuously between episodic disturbance events, building the thin-bedded fabric that characterizes the bituminous limestone. Within that fabric, organisms are preserved in intimate contact with the microbial substrate rather than scattered through a coarser sediment. Detailed sedimentological examination reveals a more dynamic picture as well, with a distinct slumping horizon and mass flow deposits interspersed within the otherwise fine-grained record. Episodic silty event beds, materials swept down from shallower parts of the surrounding platform during storms or gravity flows, played a pivotal role in the preservation chemistry.
The organism Yancyromulus zhangyi is typically found mantled by a thin veneer of fine-grained silt, the precise deposit that smothered it and ended its life, but also the physical cap that isolated its remains from further disturbance and allowed authigenic minerals to crystallize around and within its decaying body wall before structural collapse could erase the organism's outline. That pattern repeats at multiple horizons across the Shibantan. Fine material carried by these beds imposed low permeability over fossil-bearing intervals and restricted the exchange of pore fluids in ways that promoted mineral stabilization of the organic remains, an active contribution to the preservation chemistry rather than a passive burial event.
At the Gaojiashan Lagerstätte in Shanxi province, pyrite replacement operated with a chemical specificity that opens a different analytical door. The prerequisite was a sediment pore water environment in which reactive iron and bacterially generated hydrogen sulfide were both available in sufficient quantities to drive iron sulfide precipitation directly against biological structures before those structures lost their geometry. The Gaojiashan tubular fauna, Claudina, Conotubus hemiannulatus, Gaojiashania cyclus, and the various morphological types within cynnotubulites were preserved because their tube walls had enough structural coherence to guide that mineralization rather than simply collapsing into formless masses. The precision of that record yields details accessible through no other preservation mode. Claudina specimens from the Gaojiashan material sometimes display small circular perforations in their tube walls, drill holes interpreted as predatory borings, left by an unknown predator capable of targeting and penetrating a mineralized or semi-mineralized tube. Only pyrite fidelity preserves wall architecture with enough three-dimensional resolution to distinguish those borings from post-depositional damage or taphonomic artifacts, turning the preservation window into an indirect record of biotic interaction. Where compressional or mold preservation would have obliterated such fine structural evidence, the iron sulfide template retained it. The range of morphological types within cynnotubulites alone, tubes with hexagonal cross-section ornamentation, pentacarinate arrangements, and triangular geometries, is similarly recoverable from the Gaojiashan material precisely because pyrite fidelity preserved wall architecture that compressional modes would have flattened or obscured. That structural diversity, legible only through this particular taphonomic window, raises biological questions about whether these morphological variants represent distinct species, different growth stages, or environmental responses within a single taxon. Questions that remain closed without the fidelity the Gaojiashan geochemical environment happened to provide. A fifth preservation mode, operating within the broader Shaanxi and Sichuan sections, adds a dimension the other modes cannot. Where pore water chemistry at burial was enriched in dissolved phosphate rather than reactive iron and hydrogen sulfide, a different mineralization pathway became available. Calcium phosphate precipitation against soft biological surfaces, driven by bacterial decomposition of organic matter within the sediment, raising local phosphate concentrations beyond the threshold for apatite nucleation. This mode operates on a timescale fast enough to capture organisms that possessed no mineralized skeleton, no structural coherence capable of guiding authigenic crystal growth, and no organic chemistry robust enough to resist decay without immediate chemical intervention. The vase-shaped microfossils Protolagina and Sisiolagina owe their survival to exactly this speed differential. The phosphate mineral template formed before their flask-shaped tests could lose geometric integrity. The phosphatized columnar microfossils from the Zhenba section at the formation's base, whose organized architecture is interpretable as early coralline algae, survived by the same mechanism. What phosphatization contributes to the formation's taphonomic toolkit is a chemical threshold lower than any other modes, a capacity to capture organisms that every other preservation pathway in the Dengying would have systematically excluded.
The modular fossil Curviacus ediacaranus, preserved in the Shibantan bituminous limestone rather than the Gaoyoshishan, illustrates how even within a single site, a preservation process can be read backward from result to mechanism to biology. Curviacus is reconstructed as a benthic organism composed of serially arranged crescent-shaped chambers, and its preservation mode is distinctive. The chamber walls are replicated by calcispars, calcium carbonate crystals that grew within the spaces vacated as the organic chamber walls decayed, while the interiors of those chambers are filled by micritic sediment. This two-stage process required the organism's chambers to retain enough geometric integrity during early diagenesis for the growing crystals to be guided by the original wall positions rather than precipitating in an unstructured mass. The taphonomic implication is biological. Curviacus possessed a modular body construction sufficiently robust in its three-dimensional organization that the chamber geometry persisted across the critical interval between death and mineral replacement. A body plan that collapsed immediately upon death would have left nothing for those calcispars to replicate.
The annulated tubular fossil Wutubus annularis, also from the Shibantan limestone, carries a taphonomic message through its spatial relationship with bioturbation traces, including the looping burrows of Lamonte travalis. Preservation under conditions of active bioturbation is considerably more challenging than preservation in undisturbed microbial mat sediments, because burrowing organisms physically disrupt the fine-grained fabric that stabilizes organic remains and disturb the geochemical gradients that favor authigenic mineral growth. That Wutubus is preserved at all in a bioturbated context implies either that it was captured during intervals of reduced bioturbation activity, or that its semi-rigid tube walls were mechanically stable enough to resist disruption from surrounding burrowers. Some specimens preserve individual segments as internal molds, revealing that the segments were hollow rather than solid, a structural detail that only becomes apparent because the taphonomic process happened to fill those hollow interiors with fine-grained sediment before the outer wall collapsed, effectively casting the internal space and making its geometry readable. For the Zhangchuan biota, the preservation mode is carbonaceous compression, and the geological setting that produced it is as specific as any of the chemical environments described above. The fossiliferous horizon preserves organisms that were rapidly buried in fine-grained sediment in what had once been a shoreline-adjacent environment, somewhat below the low tide mark, in quiet water shielded from the energetic shallow zone nearby. This setting is broadly analogous to the depositional context invoked for the Burgess Shale, a below-tidal quiet water environment where fine particulate carbon could accumulate and organisms could be entombed before scavenging or bacterial decay erased them. The combination of rapid burial, fine grain size, and restricted oxygen in the bottom water created the conditions for carbonaceous compression, which selects for organisms with resistant organic integuments and body architectures whose structural complexity can be communicated through a flattened film. Organisms with loosely organized or chemically unstable tissues compress into ambiguous carbon smears, which means the Zhangchuan taphonomic process, despite its exceptional quality, was itself selective in ways that constrain the biological picture it provides. The relative scarcity of recognizable frondose organisms in the Zhangchuan carbonaceous compression assemblage compared to their prominence in the Shibantan limestone almost certainly reflects a genuine ecological difference between the two communities rather than a deficiency in the Zhangchuan taphonomic process. Conversely, the limited documentation of bilaterians and complex soft-bodied organisms in the Shibantan limestone compared to their abundance at Zhangchuan reflects both ecological variation and the specific selectivity of authigenic mineral preservation for organisms with structured, persistent body plans. Reading the two records together, rather than treating either as a complete portrait, allows a fuller picture of terminal Ediacaran life to emerge, and recognizing what each mode omits is as important as cataloging what it captures. The Tongshan Lagerstadt's carbonaceous preservation mode offers a diagnostic framework for this question of taphonomic selectivity. Researchers working on the Tongshan material described its assemblage as operating across 3 distinct preservation tiers simultaneously: traditional Ediacara-type preservation that records only outlines, an intermediate variant that retains organic residues with some compositional information, and full Burgess shale-type preservation that captures detailed soft tissue architecture. These tiers are populated by specific organisms rather than distributed randomly. The rangeomorph fronds, particularly the specimen with 3 distinct longitudinal regions, parallel primary branches, and perpendicular secondary branching, whose body tapers distally and curves to one side, achieve Burgess shale-type fidelity, as do Paleopycnus linearis and Eoandromeda octobrachiata. Protoconnites minor, by contrast, is represented by carbonaceous films that communicate outline without the tissue-level structural information the rangeomorphs preserve. That distribution reflects real biological differences in tissue chemistry and the concentration of resistant organic molecules in different body compartments. Differences invisible when organisms were known only through their outlines, but legible when the full taphonomic spectrum is available for comparison. The Tongshan gradient thereby transforms from a curiosity of one exceptional locality into a calibration tool for interpreting what any Ediacaran preservation mode can and cannot communicate about the organisms it holds. What those 5 taphonomic modes collectively suggest about biological information recovery is something no single preservation window can communicate alone. The fidelity ceiling is determined not by the age of the deposit or the mineralogy of the rock, but by the precise convergence of chemistry, sediment grain size, burial rate, and biological structure at the moment of death. The predatory borings on Claudina, preserved by pyrite because nothing else could have kept them, are one illustration of how far below ordinary paleontological expectation that ceiling can reach when conditions align. Those borings document not a body, but a behavior, and not a behavior alone, but a predator-prey interaction, an ecological relationship that left no independent body fossil, but was captured anyway through the structural fidelity of pyrite. If the preservation modes of the Dengying Formation are already capable of recording something as contingent and ephemeral as the act of predation, the question of what other behavioral and ecological information remains below their current detection threshold is one of the more productive open questions the formation still generates.
Chapter 5. The Ocean the Animals Inherited. The fossils described across previous chapters are not floating in isolation. Every organism preserved in the Dengying Formation lived within a specific chemical and physical environment, one that determined whether mineralized tubes could form at all, whether oxygen was available to animals that needed it, whether nutrients flowed through the water column in sufficient quantities to sustain suspension feeders, and whether the subtle shifts in ocean chemistry we can still measure in 500-million-year-old rocks were gradual enough for life to adapt or sharp enough to winnow communities down to their hardiest survivors. The geochemical archive embedded in the Dengying Formation's carbonate rocks is as remarkable, in its own way, as the fossil record it contains. Measured through the stable isotopes of carbon, oxygen, sulfur, and strontium, and through the trace element distributions preserved in well-retained limestone and dolostone, that archive documents an ocean in transition, one whose instabilities both challenged and, in some cases, enabled the biological experiments underway during this critical interval. That transition was not unique to South China. Global records document a general decrease in the magnesium to calcium ratio of seawater and a rise in dissolved carbon dioxide concentrations from the late Precambrian into the Cambrian, reflecting widespread shifts in ocean atmosphere chemistry against which the Dengying Formation's more detailed record must be read.
Start with the simplest but most consequential of the geochemical signals, ocean alkalinity. The algal dolomite member at the base of the Gaojiashan section in southern Shaanxi province preserves features that speak directly to conditions in the terminal Ediacaran ocean that would be unusual by any later standard. Within the dolomite of this member, carbonate breccias, void-filling botryoidal precipitates, and aragonite crystal fans are common structures. The aragonite crystal fans are particularly significant. Aragonite is the less thermodynamically stable polymorph of calcium carbonate, metastable under most modern ocean conditions and tending to convert to calcite over geological time. Its occurrence as an authigenic cement, precipitating directly from seawater pore fluids within the sediment, requires exceptionally high levels of carbonate saturation in the surrounding water. The terminal Ediacaran Ocean, at least as documented by the Dengying Formation's lower members, maintained a level of alkalinity substantially higher than anything typical of the Phanerozoic oceans that followed. Seawater was supersaturated enough that carbonate mineral precipitation occurred readily, filling pore spaces and void structures with aragonite cements that have since been partially converted to calcite, but whose original character is still readable in well-preserved specimens. That aragonite-forming ocean was itself a product of the globally elevated Mg-Ca ratio characteristic of late Precambrian seawater, conditions that favored aragonite and high magnesium calcite over calcite, even as those conditions were in progressive decline across the Ediacaran-Cambrian transition. This high alkalinity environment carries direct relevance for the biological story of the Dengying Formation. When seawater is already supersaturated with respect to calcium carbonate, the energetic cost of biomineralization drops substantially, and organisms that have evolved even rudimentary calcification capabilities can take advantage of the surrounding chemistry without investing heavily in complex secretory systems. The tubular organisms of the Dengying, Claudina, Cenotubulites, Conotubus, Gaugiaschenia, appear precisely when the surrounding ocean's alkalinity was primed to support mineral precipitation. Whether elevated alkalinity was a cause, an enabler, or simply a co-occurring feature of early biomineralization remains an open question. But the coincidence of these chemical conditions with the first appearance of biomineralizing animals in the stratigraphic record has attracted sustained scientific attention. The chemostratigraphic data from the Gaugiaschan member also carry information about redox conditions, and here the taphonomic context developed in Chapter 4 and the environmental analysis of this chapter converge on the same underlying chemistry. The iron sulfide reactions that drove pyrite replacement of organisms at Gaugiaschan, bacterial sulfate reduction, the generation of hydrogen sulfide, and its reaction with reactive iron, also function as an environmental proxy when considered at the scale of the whole member rather than individual fossil specimens. Elevated rates of pyrite burial and simultaneous organic carbon burial through substantial portions of the Gaugiaschan member indicate that bottom waters and shallow sediment pore waters were poorly oxygenated at best and truly anoxic across much of the basin during this interval. The strontium isotope record from the same section adds a further geochemical constraint. The 87-SR-86-SR ratio reaches a minimum of 0.7084 in limestone from the Gaugiaschan member, a value that, alongside the carbon and sulfur isotope profiles, reflects the combined contributions of continental weathering flux and hydrothermal inputs to terminal Ediacaran seawater at this location. Paradoxically, the organisms that flourished in and around this anoxic setting were precisely the tubular animals the Gaugiaschan Lagerstadt is famous for. Their presence in a low-oxygen environment may reflect several things simultaneously. Tube construction was itself potentially an adaptation to chemically challenging conditions, providing a protected microenvironment with different chemistry from the surrounding sediment. Alternatively, the organisms may have occupied a redox interface zone, living at or just above the boundary between oxygenated near-surface water and anoxic bottom conditions, where nutrient concentrations are often elevated and competition for ecological space is lower than in fully oxygenated settings. What the sulfur isotope record makes clear is that the benthic environment of the Gaugiaschan member was not the well-oxygenated seafloor that characterizes much of the Cambrian and Phanerozoic fossil record. The animals preserved there were pioneers of a challenging chemical frontier. The carbon isotope record from the Gaojiashan section introduces another dimension, and one with implications reaching well beyond the boundaries of South China. The uppermost Gaojiashan member records a major negative excursion in carbonate carbon isotopes, with values dropping to approximately -6 per mil relative to the standard marine carbonate reference. That magnitude of negative shift, and particularly its stratigraphic position immediately below the transition into the overlying Beiwan member, is comparable in both shape and magnitude to the negative carbon isotope excursion documented from Ediacaran Cambrian boundary sections around the world, a feature used repeatedly as a global correlation marker. The suggestion from the Gaojiashan data is that the Precambrian-Cambrian boundary may fall near the top of the Gaojiashan member, which would mean that the overlying Beiwan member, composed largely of dollostones and containing bitumen, represents earliest Cambrian rather than terminal Ediacaran sediment. If correct, this interpretation would make the Gaojiashan section a candidate for one of the rare shallow water sections that spans the Precambrian-Cambrian boundary in an essentially continuous carbonate sequence. And it is precisely the quality of this chemostratigraphic profile, the carbon isotope excursion documented through an uninterrupted carbonate sequence, that has led researchers to identify the section as a potential formal boundary stratotype. This matters biologically because it would mean the tubular organisms of the Gaojiashan member lived on the Ediacaran side of the carbon isotope excursion rather than straddling it. The negative excursion would then mark not simply a geochemical event, but a biological transition, with the pre-excursion Gaojiashan fauna representing a terminal Ediacaran community and the post-excursion interval representing an earliest Cambrian one. The Dengying Formation's chemostratigraphic record offers a continuous, high-resolution sequence in which to test whether changes in carbon isotope values correlate with shifts in fossil community composition, a test that requires integrating the biological and geochemical datasets at the same stratigraphic resolution, which the Gaojiashan section is particularly well positioned to support. Moving from the Shaanxi section to the Eastern Yunnan record brings a different perspective on redox conditions. Geochemical analyses of the Dengying and overlying Zhujiaqing formations in the Suzhihua section of East Yunnan have employed rare earth element distributions and cerium anomalies alongside carbon isotope profiles to reconstruct the redox state of the surrounding ocean through the Ediacaran-Cambrian transition. Cerium anomalies in carbonate rocks reflect the oxidation state of seawater at the time of deposition, because cerium behaves differently from its neighboring rare earth elements under oxic versus anoxic conditions. Negative cerium anomalies indicate oxidizing conditions, while less negative or absent anomalies suggest reducing or anoxic conditions. The East Yunnan data show that the region experienced fluctuating redox throughout the relevant interval. CE/CE* values range from 0.57 to 0.88 in the Bayan Sha member, from 0.56 to 0.83 in the Daibu member, and from 0.60 to 0.96 in the Zhangyiechan member. The upward extent of that final range, values approaching 1.0 indicating an effectively absent cerium anomaly consistent with reducing conditions, documents a progressive shift toward anoxia in the later part of the sequence, even as earlier members maintained at least intervals of suboxic to more oxygenated states. This is not a single clean transition from oxic to anoxic, but a prolonged oscillation punctuated by excursions in both directions. This spatial and temporal variability in redox conditions carries ecological implications that have received less attention than they deserve. If different parts of the Yangtze platform carbonate system were oscillating between oexic and anoxic conditions on timescales of thousands to hundreds of thousands of years, then organisms living on that platform would have been subject to repeated environmental pressures, periods of oxygen stress alternating with periods of relative chemical stability.
Such oscillating conditions have been proposed in other geological contexts as drivers of ecological innovation, selecting for organisms capable of tolerating chemical variability or exploiting the nutrient fluxes that tend to accompany redox transitions. The conidinarians, tubular biomineralizers, and bilaterians of the Dengying Formation were not evolving in a stable, uniform ocean, but in one that periodically challenged them with chemical stress, and just as periodically rewarded survivors with the nutrient-rich conditions associated with the release of metals and phosphate from anoxic sediments.
That phosphorus connection is one of the more recently illuminated aspects of the terminal Ediacaran environment. Detailed analyses of carbonate-associated phosphate, phosphate incorporated into carbonate mineral structures at the time of deposition, conducted specifically on samples from the Gaojiashan section in Shanxi province, suggest that the inorganic phosphorus reservoir in the terminal Ediacaran Ocean was substantially smaller than that of the Cambrian Ocean that followed. The data set is necessarily localized. It reflects conditions at one section of the formation rather than its full geographic extent, and extrapolating its conclusions to a basin-wide or global scale requires caution. Within its scope, however, it implies that primary productivity at the Gaojiashan site was constrained relative to later intervals. Phosphorus is a fundamental limiting nutrient for primary producers, and its availability controls the total quantity of organic matter that can be produced in surface waters. The animal communities of the Gaojiashan member were operating within a tighter ecological budget than their Cambrian successors. The transition to the Cambrian on this reading involved not only biological innovation, but also a geochemical shift toward a more productive ocean, one whose capacity to sustain larger and more energetically demanding communities was itself a product of a changed phosphorus cycle. The platform architecture shaped the ecological landscape in ways the biological record alone cannot fully reveal. In the Sichuan Basin, three-dimensional stratigraphic modeling of the Dengying Formation has demonstrated that the distribution of carbonate-producing environments was not static, but evolved systematically in response to sea level fluctuations. Carbonate-producing ecosystems in the basin thrived under specific physical parameters. Modeling indicates that dominant westerly winds from approximately 270 degrees at 80% frequency, or northerly inputs at roughly 15 degrees at 60% frequency, over an effective water depth of 10 to 20 meters, defined the productive window for mound shoal development. During periods of rising sea level, accommodation space increased. Water depth over the platform exceeded that productive range. Carbonate productivity declined, and the mound shoal complexes that hosted the most biologically active benthic communities became restricted. During intervals of falling sea level, the water shallowed back into the 10 to 20 meter range. Light penetration improved, and the complexes expanded laterally, reopening habitat for the communities that depended on them. This cyclical contraction and expansion would have repeatedly concentrated benthic life during transgressions and dispersed ecological opportunity during regressions. The Dengying Anyu Trough, a structurally controlled intracratonic depression, originated through multiple episodes of rifting in the cratonic basement, driven by the episodic tectonic uplift associated with the Tongwan movement. It began developing as the second member of the Dengying Formation completed its deposition, then deepened and widened through the later Dengying interval, until it divided the rimmed carbonate platform into two segments with somewhat different depositional histories. The depth and restricted circulation that characterized the trough would have maintained anoxic or disoxic conditions in its bottom waters even during intervals when the surrounding platform remained well oxygenated, creating a persistent redox gradient across a relatively small geographic area. That gradient, sustained over hundreds of thousands of years, maintained distinct chemical microenvironments for communities at different platform positions, preserving, in sedimentary form, a record of ecological differentiation driven not by biology alone, but by the underlying geology that shaped the basin. Reading the geochemistry and sedimentology of the Dengying Formation alongside its fossil record does not simply add context to the biological story, it constrains it. The terminal Ediacaran Ocean, as the Dengying Formation documents it, sat within a global trajectory of declining MgCa and shifting carbonate saturation, while locally oscillating between alkaline supersaturation and episodic anoxia, between phosphorus-limited productivity and the nutrient pulses that accompanied redox transitions. What the chemostratigraphic record cannot yet fully resolve is how tightly those geochemical oscillations correlate at the resolution of individual stratigraphic members and biotic horizons with the shifts in community composition visible in the biological record. That correlation between isotope excursions, trace element anomalies, and community-level turnover is the analytical frontier the Gaugian section's combination of stratigraphic completeness and preserved geochemical signal is uniquely equipped to address. The Dengying Formation stands as far more than a catalog of ancient organisms. It is a reminder that the story of life's complexity does not begin where our records become clear. It begins earlier, in environments we have only begun to learn how to read. The Zhongshan biota shows us that animals we call Cambrian were already living, feeding, and thriving in Ediacaran seas. The Tangshan Lagerstadt shows us that what we call extinction may sometimes be nothing more than silence in the rock. And the geochemistry shows us that biology and ocean chemistry were locked in a conversation whose full vocabulary we are still decoding. Every new exposure, every weathered outcrop, potentially holds evidence that rewrites our understanding of when complexity first took hold on Earth. The greatest finds in this formation may still be waiting. Thank you for listening.